Awesome. Thank you once again, Gerhard. So our next speaker we have with us is Dr. G.A. Nagana Gowda, the Research Associate Professor at University of Washington. Dr. G.A. Nagana Gowda is a Research Associate Professor at the Northwest Metabolomics Research Center, Department of Anesthesiology and Pain Medicine, University of Washington, Seattle. From 2006 to 2012, he was a research scientist in the chemistry department, Purdue University, West Lafayette, Indiana. Previously, he was an assistant professor at the Center of Biomedical Magnetic Resonance, Lucknow, India. He did his postdoc work at the State University of New York, Buffalo, and worked at the NMR Research Center, Indian Institute of Science in Bangalore, India. He received his bachelor's degree in 1983 and his master's degree in 1985 from Mysore University, India, and his doctoral degree in NMR spectroscopy in 1999 from Bangalore University, India. Broadly, his research is in the area of NMR spectroscopy-based metabolomics, methods development and applications focusing mechanistic insights into health and pathophysiology of diseases. His talk today is entitled Isotope Labeling and NMR Spectroscopy-Based Metabolomics. And you should be able to go ahead and share your screen. Thank you for being present with us today. I believe you are on mute.
Okay, how about now? Can you hear me?
We can hear you now.
Okay, can you see my slides?
Yes, we can.
Okay, good afternoon, everybody. A little bit about introduction. Metabolomics is all about analysis of small molecules in biological samples focused on understanding human health and diseases. The major steps involved in metabolomics is analysis of samples, detection, identification, and quantitation of the metabolites. They use these metabolite concentration for disease diagnosis or focused on mechanistic understanding and then identifying drug targets. Isotope labeling is a major component in metabolomic research. We basically use in three areas. One of them is internal standards for metabolite quantitation, and then we use metabolite profiling, combining isotope tagging, and the third one is pathway and then flux analysis. Quantitation purposes, use of isotope labeling is basically the mass spectrometry area. We just use a mixture of isotope labeled compounds in a mix with samples and then detect both unlabeled and labeled compounds, then determine the concentration of the metabolites. We use about 40 metabolites, isotope labeled compounds currently. Unfortunately, we don't have isotope labeled compounds for all the metabolites that we analyze. Hundreds of metabolites we typically analyze. This is the most recently in the process of developing a method for the analysis of glutathione in biological samples. We discovered a method to isotopically label glutathione in biological samples, depending on the solvents that we use, whether it's water or deuterium. We use N-ethyl maleimide to tag glutathione because glutathione is extremely unstable compounds. So we end up with getting about 12 different deuterium isotopomers for glutathione. So the use of this is you can use this as a quantitative standards in mass spectrometry analysis. Is a very simple way of generating this isotope labeled compound. So other major area where we use isotope labeling is for tagging metabolites in biological samples. As you know that because of NMR's limited sensitivity and resolution, so the number of metabolites detected is far few compared to mass spectrometry. So one remedy is to tag metabolites using isotope tag and then detect using 1D or 2D NMR, so that you get to identify detect more metabolites. You can significantly enhance the pool of metabolites that you can detect. So unfortunately, you cannot tag all the metabolites present in the biological samples. You have to go by class by class. For example, so a main class of metabolites, you just tag using 13C labeled acetic anhydride, for example. Then nitrogen labeled tag, ethanolamine that we use for carboxylic acid class of metabolites detection. Then hydroxyl group containing metabolites, we use phosphorus tag for detection in biological samples. This is just an example of tagging carboxylic acid class of metabolites in biological samples using nitrogen labeled compound. So we detect about 200 peaks in the nitrogen 15 proton HSQC spectrum for human urine, which is equivalent to roughly 200 metabolites, which is unprecedented for NMR, so detection of so many metabolites in a single experiments. This is a similarly another tag. We use the nitrogen 15 phthalimide to form to first synthesize a tag called smart isotope tag, which has cholamine and 15 cholamine. We call it smart tag because it has got permanent charge as well as nitrogen 15 labels, so you can detect NMR spectroscopy and high sensitivity. You can also detect by mass spectrometry with high sensitivity because of its permanent charge. So what you see here is an examples of serum and urine spectrum tagged after tagging with this smart cholamine tag. So the third area where we use extensively isotope labeling is for metabolic pathway analysis, because as you know, this isotope tagging lets you distinguish metabolites from different pathways. You can analyze pathways and then measure flux analysis. So there are two ways under which we do this analysis. One of them is in vitro labeling and then ex vivo analysis. The other one is in vitro labeling and then in vitro analysis. So this in vitro labeling ex vivo analysis, we just feed cells or subcellular organelles, animal models, or humans with isotope tag, and then extract the samples, then analyze using NMR spectroscopy. Analysis of pathway using NMR spectroscopy is unlike mass spectrometry, is interesting in that here you will not only get to know which metabolite receives this tag, and you will also know which atom in the metabolite receives the tag. So based on the NMR signal from one particular atom or nuclei, you'll get to know the information about multiple pathways. For example, if you feed glucose, 1,6-13C labeled glucose, so it forms one of the carbon is isotopically labeled in acetyl coenzyme A. So as a result, you get glutamic acid where number 4 carbon is labeled. So when it undergoes, so you get signals. If you measure signal for number 4 carbon, is 13C NMR, you get singlet. Similarly, when it undergoes multiple cycles, so you get another glutamic acid is 3,4 labeled, and for this you will see doublet. Similarly, instead of glucose, if you use 31 to 13C labeled acetate, then acetyl coenzyme A, both the carbons are isotopically labeled. So you get glutamic acid where 4 and 5 carbons are isotopically labeled, but you still measure carbon 13-4 number carbon atom number 4, you get doublet, which is different doublet than what you saw before. So when this undergoes multiple cycle TCA cycles, it forms glutamic acid where 3, 4, 5 carbon atoms are labeled. So for this you get 4 lines quoted. So now if you feed simultaneously acetate as well as glucose, then you will form 2 kinds of acetyl coenzyme A. So in the first cycle we have 2 glutamic acid labeled molecules. In the multiple cycles you have 2 more glutamic acid differently labeled molecules. So the net result is if you measure 13C of 4, so you guess singlet, doublet, 2 doublets, and then quadrets. So this is an example of a real sample. This is non-tumor tissue. This is a pattern of the signal for where 13C labeled from acetate as well as glucose. And when you do the experiments for tumor samples, so you get this kind of patterns. The pattern of labeling is different. So if you quantify these peaks, and then you will get to know that glucose is consumed more in non-tumor compared to acetate, whereas if you use tumor tissue, you have acetate is consumed more. So the glucose oxidation is significantly reduced in tumors. So this is what the information you get by multiple isotope tracers feeding and then identifying a pathway based on traces. So you can also identify whether the substrate goes through a PDH because it gives a unique set of signals, or whether it goes through pyruvate carboxylase. So because you get the isotopic pattern is going to be different. Similarly, you can also distinguish between a pentose phosphate pathway and then glycolysis based on tracer analysis using glucose. In this case, 1,2,13C labeled glucose if you feed. So one of the end products is lactate, where 2 and 3 are 13C labeled. So if the glucose passes through pentose phosphate pathways, then the end product is going to be a lactate, where only carbon number 3 is labeled. So NMR spectrum is going to be a singlet if the glucose goes through pentose phosphate pathway, or it's going to be a doublet because there are 2 carbons labeled if the glucose goes through glycolysis pathway. So this is an example of one of our studies where we have the bottom one is wild type cells, and the top one is PPX, that's a pyrophosphate deficient enzyme rich cells. So 1,2 glucose 13C labeled glucose was fed to the cells. You can see significantly increased glycolysis as well as pentose phosphate pathway in the PPX cells compared to wild type cells. So here is an this is for in vitro labeling and then in vitro analysis. This is analysis of live systems in real time. So whether it can be for subcellular organelles or cells or isolated organs or animal models or humans, which are fed with isotope labeled substrates and then monitored in living systems using NMR spectroscopy. This is an example of one of our studies where we have used mitochondria, isolated mitochondria from C2C12 cells. It's the mouse muscle cells. So we feed the cells, we take the samples, isolated mitochondria in an NMR tube, and then we feed them with 13C labeled pyruvate, and then we monitor pyruvate metabolism in the TCA cycle, essentially inside mitochondria. So the spectrum, what you see here on the left side is the spectrum, 2D spectrum, HSQ spectrum obtained after 10 minutes after adding pyruvate into the mitochondrial solution. This is after we record spectrum every 5 minutes because sensitivity is very high because we monitor 30C labeled substrates. In 5, you can see that alanine is tiny. 40 minutes, it just grows big. Lactate is a tiny peak in 10 minutes, becomes big in 40 minutes. Whereas pyruvate is a substrate that you have fed the cells, so it goes down in intensities. So if you measure the intensities of these metabolites, you get this is time course of some of the metabolites that we have. This is one of our recent experiments. You can see that pyruvate consumption goes down with time because it is used up, and then many metabolites intensity increase with time. So this is an experiment where we have for this mitochondria which are fed with pyruvate. We add inhibitors of pyruvate metabolism. The red one is a control, and then a blue one graph is for inhibitors added intensities of different metabolites. This is a close-up view of the same thing. What you can see here is when you do add FCCP, the intensities of the P coenzyme A increases. I mean, this is an inhibitor of mitochondrial respiration electron transport chain. So it increases oxygen consumption and increases acetyl coenzyme synthesis. Whereas if you add rotenine, it decreases acetyl coenzyme synthesis because it is known to disrupt, it is shut down the electron transport chain. So these are some of the examples. Just to summarize, isotope labeling is an important aspect in metabolomic research, particularly for quantitation of metabolic pathway. It is indispensable tool. The standards are for quantitation purposes. And isotope label standards are gold standards for quantitation. The one challenge is we don't have labeled standards for all the metabolites that we analyze. Hopefully, we will they will be available soon so that we can expand the studies of metabolomics using isotope labeled standards. With this, I'd like to acknowledge people who are associated with studies. I'd like to thank you for your attention. Any questions?
We do have one question in the chat, which is what strategies or tools do you use to confidently identify and quantify unknown metabolites in profiling exercises by NMR? Is cross-check to MS method results included?
Okay, we do unknown identification in our laboratory a lot. So we use a combination of 1D and 2D NMR techniques, and then we start with using the there's many databases. We get to know roughly what it could be. Then we start using confirming using 1D 2D NMR experiments, and then finally we do a spiking using authentic content compounds to make sure that it is the correct compound. We do a lot of unknown identification in our laboratory routinely.
Awesome. Thank you so much for that explanation and for this wonderful presentation.
Thank you.
Alrighty. Our next